video. Has all the details in there. And Bozeman Science in general is a really good resource for reviewing. I do highly recommend it. So we'll go over photosynthesis, but we've talked about all the important components, so we'll kind of just go over it on the surface level, and then we'll talk about cellular respiration. So again, photosynthesis occurs in only certain organisms, such as plants, some bacteria, some protists, including algae. So a lot of organisms that appear to be green, algae, some protists, those tiny organisms, tiny eukaryotes, and blue-green cyanobacteria. And all of these organisms that perform photosynthesis have something in common, and that is that they have these pigments which absorb light energy. In plant cells specifically, these pigments are stored in these certain organelles, which are called chloroplasts. And the chloroplasts have multiple membranes that separate these reactions. So why are these reactions, or how are these reactions separated? Well, we have multiple compartments separated by membranes, and then enzymes are only in certain compartments. That the reactions only occur in certain compartments. So here we have an image of the plant cell and a chloroplast, which is where the enzymes for photosynthesis are housed in plant cells. So if we're talking about bacteria, which are prokaryotes, do they have chloroplasts to house photosynthesis? Do bacteria that perform photosynthesis also have photosynthesis going on in these same structures? So do bacteria also have chloroplasts? No. And how do we know that? Yeah. They're prokaryotes, so they don't have membrane-bound organelles. So bacteria will have photosynthesis going on inside of the cell in general, and some of the enzymes that have to be in a membrane are going to be in the cell membrane. And so this is an image showing us that our chloroplasts actually have, so I should have deleted these arrows. So this is the whole cell, and then these are chloroplasts within the cell, and we can see that they're green because, again, they have that chlorophyll in them. And this is the absorption spectrum that we saw in the video, which again shows that the green light is actually reflected and the other colors of light are absorbed. So tomorrow in lab, we'll see that plants contain various pigments, and some of them will absorb different wavelengths of light. And the different pigments overlap and kind of help one another. So certain pigments are going to absorb best in the blue range or purple range, and then some pigments are really good at absorbing in the red range. And so by having multiple pigments, the plants could absorb more energy because the different pigments are absorbing best at the different wavelengths. So it would be kind of like we saw different enzymes working at different temperatures. So it would be like having a bunch of enzymes that all work at different temperatures, and then exposing those enzymes to high temperatures, medium temperatures, and low temperatures, and then you constantly have some of the product being made. And so this is just an outline. Again, we're turning light energy into bond energy through the absorption of the light energy by an excited electron. And something that I wanted to point out that I'm not sure if really mentioned in the video is why do we go through this complicated set of reactions to go from light energy into bond energy? Well, in the big picture, it's because the light energy that is absorbed contains a lot of energy. So the photons that are absorbed initially by the chlorophyll are very high energy. And so I show this GIF because we see that the photons that are absorbed are very high energy, and essentially we couldn't store all that energy in one molecule. So there's a process where multiple molecules are going to get each a little bit of the energy from this photon. So this is a really nice image showing, again, a review of what's going on here. So on the left side, we have light energy coming in, and it's absorbed by the specific complex, which is called the antenna complex. And this, again, houses the pigment. This is where the electron is actually going to be excited. And again, that electron comes from what molecule? Water. So we also have, as part of this antenna complex, the water splitting enzyme. And the water splitting enzyme is crucial because if we didn't have that, then we would just have photons coming in and they wouldn't be absorbed by anything. So just like we're not getting any energy from standing in the sunshine or standing under the light right now, the same thing would happen if we didn't have this water splitting enzyme. There would be nothing to actually harness the energy from the photon of light. But luckily we do have this enzyme. Then this electron is passed through between various enzymes. And you don't need to know the name of all of these, but I did want to mention this type of enzyme. So this is a cytochrome. Does this ring a bell for anyone? Maybe a little bit. Cytochromes. You may have heard of them before. So cytochromes are enzymes that can bind to electrons and they pump protons. They're situated in membranes, and they are also an ancient type of enzyme. So plants have them, bacteria have them, humans and mammals have them, and they're all different types of cytochromes that have been slightly modified throughout time, throughout evolutionary history, but they all do a similar job. They bind to electrons, they use a little bit of that energy to then pump a proton across a membrane, no matter what organism they're in. And then we see, remember we have two sets of reactions. We have photosystem 2, and then we have photosystem 1. So this first part, of course, is photosystem 2. Then we're moving into photosystem 1. After these protons are pumped, the electron is passed on to some other enzymes, and we get the electron re-energized. And so it goes back up to a higher energy, and then it's passed on between more enzymes that are attached to the membrane. One of the products of photosystem 1 is NADPH. So you go from the lower energy form of the molecule, NADP+, and you add on a hydrogen to make NADPH. So that's NADP+. Add on proton. This is activated ion. activated form, this is the low form of the molecule. So this reaction in this direction, do we think that this has a positive or a negative value for delta G? Talk to the person next to you for 10 seconds. Positive or negative? So how many people think, raise your hand if you think that the delta G is going to be positive? Okay. How many think that the delta G is going to be negative? Okay, so this delta G for this reaction is indeed positive because this reaction requires energy. An energy requiring reaction because we go from a low energy molecule to a high energy molecule. And where does the energy for this reaction come from? What are we passing between enzymes? Electron, right. So we're taking some of the energy from that electron and we're using it to change the shape of the enzyme so that it adds the proton onto NADP to make NADPH. All right, so back up to the slide here. And NADPH, as we see, is kind of left hanging here because we're going to use it in the next set of reactions. So again, this is one of the products from photosystem 1. The very end of photosystem 1 involves this enzyme called ATP synthase. This is the most effective way or most prolific way to make ATP in the cell. So most of the ATP in our cells comes from this enzyme, and most of the ATP in plant cells comes from this enzyme. So again, I give you a hint there that this is a very old enzyme. It's very important. Both plants and animals have it, and bacteria. So ATP synthase is the only way in which protons can move back across the membrane. So as we discussed earlier in the week, according to the laws of diffusion, these protons are going to want to cross back over the membrane because there's a high concentration of protons in the thylakoid space here. There's a low concentration of protons in the stroma. But the only way for protons to pass through the membrane, because they're ions and they can't get through the membrane, they can't diffuse through spontaneously, they need to go through ATP synthase. And this enzyme actually works like a little machine. So it's actually referred to as a molecular machine. And it binds to a proton, and there's a certain part on ATP synthase that turns as it binds to protons. So it binds to a proton, turns a little bit, binds to another proton, turns more. And as one piece of the enzyme turns, the other part of the enzyme catalyzes the reaction to add a phosphate group onto ATP. So it's like a little machine that's powered by a crank. And again, the power is coming from the protons moving through it. And then it's using that to add a phosphate group onto ADP. Yeah, don't worry, I'm not erasing it. So ATP synthase. It binds to ADP, again, the lower energy version of the molecule, plus an inorganic phosphate. It makes ATP in water, high energy. And what fuels this reaction is H plus concentration gradient, which causes the turning of something called a rotor, which is part of the enzyme. And again, that physical turning and the chemical turning of that part of the enzyme is what actually makes the reaction go forward.
There's another
This is the enzyme that adds phosphate. ATP synthase. And again, ATP synthase is an enzyme that you find in plants.
It's 11 o'clock.
Bacteria. It's 11 o'clock. And ATP synthase is always found in a membrane, because if it's not found in a membrane, then why would protons go through it? You have to have ATP synthase in a membrane, and you have to have a concentration gradient. Otherwise, protons are not going to go through it. And similarly, just like for a couple other things we talked about today, this reaction could go in either direction. So this enzyme can actually use ATP to move protons in the opposite direction. And this does occur sometimes if, for example, the rest of the electron transport chain isn't happening fast enough, if there's damage to the membrane, any reason why that proton gradient isn't set up in the correct way, then ATP synthase can use ATP to pump protons in the opposite direction. Obviously, not ideally what we would like to happen, because we want to be using that proton gradient to make ATP, not using ATP to set up the proton gradient. And so in the big picture here, for photosynthesis, and we'll see for the end of cellular respiration, we have a lot of steps. One of the main goals is to set up a proton gradient so that ATP synthase can use it. So there's a lot of steps to get protons in a certain place. Seems kind of ridiculous, but hey, it works. Then what happens after this? We talked briefly, or the video talked about what occurs after this. Where does NADPH go? The Calvin cycle, right. And in the Calvin cycle, that is called carbon fixation. Because we can't just make a bunch of ATP. That's not a basis for life. We also need carbon-based molecules. Including things like glucose. And so that's going to be our transition into cellular respiration. So we thought that this was a nice little diagram because it shows the connection between the chloroplast and the mitochondria. So the chloroplast is the site of photosynthesis in plants and algae. And the mitochondria is the site of cellular respiration. And so these two sets of reactions are linked. And also, as a side note, we can see that there are a lot of similarities in structure between the chloroplast and the mitochondria. There are both membrane-bound organelles that are roughly the same size-ish, and they both have a double membrane, and we think that this is because both of them originated as bacteria that were then endocytosed by some eukaryotic cell. And then these bacteria ended up surviving and living in symbiosis with the eukaryotic cell. And both of these organelles, chloroplast and mitochondria, we kind of gloss over their replication when we talk about mitosis, right? We didn't really talk about that. That's because they replicate independently of the rest of the cell, because they're kind of still, they're like not fully domesticated. They both actually have their own genomes. They're really small, they don't have all the genes to live on their own, but they do have their own set of genes that specify all the enzymes that are needed for photosynthesis and cellular respiration. So that's kind of neat. And if you take the mitochondria out of a cell, for example, it will still catalyze the reactions that it's supposed to for a little while before it runs out of resources. So here we see that for photosynthesis, again, we use water, we use CO2, and we produce O2 as well as sugars, which are carbon-based molecules. There are other carbon-based molecules as well, obviously, but we're particularly interested in sugars because sugars such as glucose are the input for cellular respiration as well as oxygen. And from cellular respiration, we get energy in the form of ATP, and the waste products are carbon dioxide and water. So you can see kind of not only how these two organelles work in concert in a plant cell, but also how ecosystems kind of depend on different members. So again, plants are the only thing that can turn, or plants and bacteria, some protists are the only thing that can really turn light energy into chemical energy. Without them, we literally would have no food on Earth. So we're going to go through cellular respiration. It has some different steps, different stages. The big picture is that cellular respiration is breaking down glucose and making ATP. And the formula for glucose is C6H12. different steps or different stages for cellular respiration are glycolysis, aerobic processing, TCA cycle, and then finally the electron transport chain. Again, we have the different stages of cellular respiration being glycolysis, pyruvate processing, the TCA cycle, which is also known as the Krebs cycle, and then the electron transport chain at the end, which is very similar to the electron transport chain in photosynthesis. So up here you see glycolysis. This is where you start out with glucose, and we're going to be breaking down glucose. Now, as I've written up here on the board, we have 2 stages to glycolysis, which are the preparatory phase and the payoff phase. So in the preparatory phase, this is a series of reactions that prepare glucose to be broken down. So it actually requires an input of energy. Series of reactions. So we actually do have to use ATP for the preparatory phases, or the preparatory stage. But then in the payoff stage, we have a series of reactions as well. And in this set of reactions, we're going to generate ATP. And we're also going to generate molecules that are going to go into the next step. So we're going to generate pyruvate. As well as so glycolysis is actually a series of 10 reactions total, split up into these 2 phases, preparatory and payoff. The first reaction we've already talked about actually. This is adding that phosphate group onto glucose. And so that is part of the preparatory stage. That reaction, that first reaction, actually uses ATP because we need to take that phosphate and put it on glucose. And the other important thing about that reaction, as we said, is that it prevents glucose from binding the glucose transporter and leaving the cell. Sorry? So it prevents glucose from binding to the glucose transporter and leaving the cell, because it's no longer glucose, it's glucose 1-P, glucose 1-phosphate. So I'm not going to talk about all 10 reactions. But again, if you are a very visual person, I personally find that it helped me just to follow through the reactions to see that this is really, there's no mystery. It's just taking all of the atoms in glucose, rearranging them, sometimes taking a few away, sometimes adding a few in form of phosphate. And at one step, also break the glucose into two molecules. So that's the other thing that happens here. We start out with glucose with 6 carbons, and then we make it into pyruvate, which is a 3-carbon molecule, but we have 2 of them. So we've broken glucose in half. Each half has 3 carbons. And the form of glucose essentially that comes out of glycolysis is pyruvate. So that's the carbon molecule, it's a 3-carbon molecule that comes out of glycolysis. So again, glycolysis does require ATP, so it actually requires 2 ATP. And it produces 4 ATP. So we do gain, we have a net gain of 2 ATP. It's not that much. It's like that meme, like it's not much, but it's not a sliver. And again, think about that. Producing net only 2 ATPs, not that much. Where we make our major gains in ATP synthesis, again, are through ATP synthase. And we also generate NADH because some of these reactions are going to add on a hydrogen to NAD. So we also require NAD plus for the input of glycolysis. And so this is again a low energy form of an energy carrier molecule. Okay, so what happens to the carbons? What happens to pyruvate after glycolysis? We have something called pyruvate processing, and you see here glucose during glycolysis is split up into 2 pyruvate molecules, and now it enters the mitochondria. And when it enters the mitochondria, then we actually convert pyruvate into acetyl-CoA. And we're losing 1 more carbon. So during pyruvate processing, we are releasing 1 carbon in the form of carbon dioxide, and we're making acetyl-CoA. And this has 2 carbons. So for most of this process, we want to be following how many carbons we have left from glucose. And eventually all the carbons from glucose, this 6-carbon molecule, are going to end up in CO2. It just takes a while. So this is where we make our first CO2 going from pyruvate into acetyl-CoA. And we can see in this image here Pyruvate has the 3 carbons, right? One in the carboxyl, one bound to oxygen in the middle, and then a methyl group. And then when we have, we only bring 2 of the carbons over, so we split them all through here. We have the methyl group and this other middle carbon, so methyl group and middle carbon, and then this carbon on the end we use as CO2. And we have that happen 2 times per glucose molecule because we have 2 pyruvate molecules per glucose. So the next phase is the TCA cycle. It's also called the Krebs cycle. It's also called the citric acid cycle. So which version were you guys told when you originally learned this? Krebs and citric. Okay. So both of those are fine. TCA actually stands for tri-R acid. You could know it by any of the names. When I put this on any kind of assignment or assessment, I try and write all 3 names because I know that they're kind of equal used. Now continuing to follow that theme that we've been talking about all day, is that we have these metabolic pathways where the reactions use the products from the previous reaction. So we're using the product from the previous step to acetyl-CoA. So this 2-carbon thing. And this 2-carbon thing then donates the carbons to this cycle. So here we have acetyl-CoA. You see that we have carbon in the middle and then methyl, and we break this bond here. So this acetyl is part of the molecule, it's just recycled. The carbons are all that are passed on to this cycle. So in this cycle, there are a bunch of different enzymes. So we have 8 different steps, 8 different enzymes. And in this process, we break down this carbon molecule even more. The tricky part about this is that this is a cycle. So we are recycling the carbon skeleton. And we're actually going to combine it with the carbon skeleton from previous glucose molecules. So in this cycle, we already have, when we start, this molecule called oxaloacetate. And this is a 4-carbon molecule. Then when we donate these 2 carbons, we have a 6-carbon molecule called citrate, which is why this is called the citric acid cycle. So once we have citrate, that's when we start to break down this molecule. We generate through various reactions NADH. We also generate CO2. So remember that in any reaction where you're generating NADH, you also usually generate CO2. And we generate a little bit of GTP, 1 GTP molecule, again, as an energy carrier molecule, it's just useful. More FADH2, so it's very similar to NADH. 1 less NADH here. So we started with 2 carbons, combined it with 4 carbons to make 6. Then that 6-carbon molecule lost 1 carbon in carbon dioxide. We have 5 carbons left. We use another carbon through carbon dioxide with 4 carbons left. and then that 4-carbon molecule goes around and is combined with the next acetyl-CoA. Do we have any questions? We usually have somebody says, how does the cycle start? This doesn't make sense. How do we have a 4-carbon molecule that started out with 2 carbons? Well, this is just showing the cycle isolated. Remember a couple minutes ago, or a little while ago, we saw that giant network, metabolic network. So this cycle connects to a lot of other reactions as well, including nucleotide synthesis and amino acid synthesis, so that you could take some of these carbon molecules and then make them into nucleotides or amino acids, or you could take amino acids or nucleotides and put them in here so that you could get a 5-carbon molecule or a 4-carbon molecule. And that's how we can into the cycle and then combine them with our target glucose. So again, that's just the big picture. You don't need to know that. It's just that we start to overthink this thing and say, I don't understand how this started. That's how it starts. Okay, so at this point, this is a dead end for our carbons, right? So we have, this is accounting for all the carbons that we started out with in glucose. We released 1 in pyruvate processing, and then we released 2 in pyruvate processing, and then we released 2 here in the CO2, and then the rest gets recycled through the cycle. So that accounts for all the carbons. And so we start to realize, okay, so the carbons are important, but only to fuel these other reactions. So what do we have that we produce that's really valuable? Things like NADH and FADH2. So we have 2 that were released during pyruvate processing, and then so we have 3 we split up 2 molecules in pyruvate, so that's all 6. Then each of these loses 1 during pyruvate processing. So then 1's lost to CO2 times 2, so that makes 2. And then we have the rest of the 4 in acetyl-CoA. Right? And then they each go into the Krebs cycle. Or we have 2 of these glucose in the Krebs cycle. 1, 2, which receives times 2, which is 4. And then the other 2 were lost less, which is and then there is a CO2 here and CO2 here. So that's again 1, 2 times 2, which is 4. Right, so this is 4 that make it into the Krebs cycle. The other 2 were released during pyruvate processing of CO2. So we've accounted for all of our carbons. Is there a question? Yes, the Krebs cycle essentially happens twice. There are 2 acetyl-CoAs, right? And both of them need to go through this process. So at this point, in total, we have made 6 carbon dioxide molecules, which you release in the next step. We have made only 4 ATP. plus the two that we had to put into glycolysis. We made lots of NADH and we made a few FADH2s. So from the whole process of cellular respiration, we usually make around 32 ATP. At the point of the TCA cycle, we've only made around 4, between 4 and 6. So where do the rest of them come from? The electron transport chain, and specifically from ATP synthase. Okay, so this is where we start to get the real payoff. So again, what are we doing? What do we have left? The only thing we have left are these molecules. We don't have any carbon molecules left, but we do have these energy carrier molecules. And what are they carrying specifically that's really useful to us since we want to power ATP synthase? We're carrying the hydrogens. So these two molecules, NADH and FADH2, are going to bring their protons to the electron transport chain. And they're going to donate these protons to the electron transport chain, and these molecules or these ions are going to be passed through the electron transport chain, just like we saw in photosynthesis, and they're going to fuel a whole bunch of enzymes that are going to pump protons across the membrane. And then we know what happens after that. Rosie?
So the hydrogens basically
They have what?
the energy.
Yes. The hydrogens have the energy at this point. And this GIF shows that NADH comes in and is going to donate hydrogen. NADH goes into Na plus. This first enzyme complex is going to bind that hydrogen, and it actually pumps the proton across the membrane. The electron gets passed on to the next protein. So the electron, no, so that was only in photosynthesis. The electrons, our electrons that get passed through the electron transport chain are not involved. Just the fact that there is no electrons, and the direct donator of the electron transport chain for our cellular respiration comes from NADH or FADH. The electron. Or NADH.
Oh, so it supplies the hydrogen.
Yes. So the proton and the electron. So again, we see that hydrogen is donated. The proton from the hydrogen is going across. The electron from the hydrogen is passed on. And then the electron that's passed on, a little bit of energy from that electron is used by other enzymes to also pump protons. So we see that one of these enzymes is actually cytochrome C. So this is another cytochrome enzyme. We talked about before, it's an enzyme carrier. And in the electron transport chain, or maybe, no, I do have a slide for this. In the electron transport chain, what we're seeing is that the electron starts at a high energy, and as it is passed between complexes, it's going to lose energy each time. So it gets this enzyme complex 1, then it gets passed to enzyme complex 2, it has lost some energy, then that energy, more energy is going to be used again, every time it gets passed between enzyme complexes, it is going to be used to fuel some kind of shape change in the protein, as we talked about before. Some of those shape changes end up pumping protons across the membrane. Some of them do not. Some of these proteins do not pump protons. But ultimately, the proteins that don't pump protons are important because they're able to carry the electron to the next protein that is going to pump the protons. And again, just like we said in photosynthesis, the importance here is that, hey, we're using a whole bunch of enzymes and we're using a lot of energy to end up making a hydrogen or a proton gradient. And the proton gradient is important because why? ATP synthase. That's where we're going to get most of our ATP generated. Also, before we go back to ATP synthase, the final molecule that accepts the electron that is at its lowest energy state is oxygen, and it's going to produce water. So this is why we need to intake oxygen, because we need it at a molecular level to bind to this electron. If we don't have oxygen to bind this electron, then basically we're causing a traffic jam, we're causing a backup, and the electron transport chain can't work. And this is why we breathe out water, because we're actually producing water from the oxygen. So again, ATP synthase uses those protons to make ATP. And this is a nice little movie showing that we have lots of protons on this side of the membrane. The ATP synthase actually binds the protons. So we can see that there are protons that are bound here in the middle, and this region actually spins around. It rotates a little bit as every one of these protons is bound. And then as it rotates, it's going to release one of the protons that was bound before. And as it's spinning, we're also producing ATP via this region of the enzyme. So this whole thing here, the brown part and the green part in the membrane here, and the yellow and red parts, and the purple and pink parts, this is all ATP synthase. It's a pretty large enzyme, and it has different components. So this is all in the mitochondria. Yes, so this is, I think that there's a better image of it. So this is inside of the inner mitochondrial membrane.
So like in that picture, what was the
right, and so
protons?
Yes, down below would be this area, which is mitochondria. Membrane 1 and membrane 2, and then all the protons are pumped into this area in between the intramembrane space. And the protons are pumped there, and then they flow into the interior, the inner compartment of the mitochondria. So this area here is in the intramembrane space, and then this is the mitochondria, is actually the very center of the mitochondria within both of the membranes. Yeah, and it really helps to, I think that this picture really helps to orient yourself when you're trying to think about it. So what questions? We talked about a lot of stuff.
Sometimes that's good.
Yeah, so it depends on the conditions. Between 30 or 32 is optimal. It also depends on the organism because the ATP synthase enzymes are slightly different. Again, this is nice. Image shows the NADH moving the electron, proton pumped across the membrane. That proton is being brought back across the membrane by ATP synthase. So do any of the carbons from glucose end up in ATP? Right, so none of the carbons, none of the atoms in glucose end up in ATP. It's important to remember. The carbons in glucose are used to fuel processes to make other molecules that are going to end up making a proton gradient that then makes ATP. Proton. So, glucose, carbons, fuel reactions. They things like NADH, H2, some of these activated energy carrier molecules, which are then going to make protons.
So, there's some slides that you is not going to H2 exist as
Let's slide what process are we talking about? What are you talking about in photosynthesis?
It's after cellular development.
Oh, yes. So actually, that's just a typo. It should say FADH2. I got my formatting got messed up with the subscript. So FADH2 is the energy carrier molecule, as we see down here. FADH2. So, non-activated molecule, again, is NADH. It's just happening. Any other questions? I know this is a lot. That's why this will not be on the quiz tomorrow. The quiz tomorrow will just be on Mondays and Tuesday stuff. And then you'll have a weekend to process this. Any other questions?
So, this process like that first process needs is used in this process.
Not everything, but some key components. Yeah, and that's, I think that is a really good way to learn this, is to try and name all of the products of each process and which ones go to the next step. And for steps 1 or phase 1, 2, and 3, we're talking about following carbons. And then for 4, we're talking about seeing where the protons came from. The protons come from all the different steps, all the different phases.